Center for Integrative Neuroscience.
I did.
And his talk will be about investigation of vision restoration through optogenetics. Take it away.
So yesterday, Yuling asked me, why do I have a picture of a tree on the cover slide for my presentation? I tried to convince him that this was actually a high-resolution photo of a neuron. And this was the axon, and these were the dendritic branches all over the screen. But what this got me to do was think about why I do have a picture of a tree on the cover slide of my presentation. And I think it's because I want to share with you the importance of metaphorical thinking, that perhaps we can think of this tree as a neuron, and if I were to study how this tree grows, how its branches actually branch, perhaps I could gain something from that and learn about how actual neurons grow and branch. And this metaphorical thinking, I think, is unique to humans, and it's created a lot of the great discoveries in science, and it makes me think. Recently I read I read an article about a 13-year-old who was hiking, and he looked up to the trees and saw how the branches spiral, and he thought, trees need sunlight. And so he actually designed a solar panel based on this, and it actually collects more sunlight than a normal solar panel. And then the same metaphorical thinking, in 1980, a guy named Ken Foster was studying algae and how they receive light. Now, what we have from this is something called channel rhodopsin. We have optogenetics. We have taken this channel out of this algae, and now we're putting it into cells in the brain, into the eye. So again, I just want to stress the importance of this metaphorical thinking. And in my study, I'll be using mice. But at the end of the day, I want to cure blindness in humans. Now, mice vision and human vision, though it might be similar, it's not the same. So what I hope to do is draw conclusions from these experiments that can be used later in the future. And these experiments totally would not be where they are today without these three people right here. I'm working with Thomas Munch in his lab, and he's a great supervisor, been with it the whole way, supporting and all of this. And Marion has also been a tremendous help. When I met with Thomas and Marion back in July after the Haile Kreuztal retreat, I had this idea for the project, and they were totally supportive and on board, and even started doing pilots back in July. And we have a saying in the lab: if we have a problem, ask Boris, and he has the answer for us. So he's been a great help too, designing the paradigm and everything. So without further ado, let's dive in. My talk is 3 parts. So the first part is what is optogenetics, just to kind of get you going. The second part is the research, and the third part is the future. A lot of this project was designed in mind as also a master's project, so a lot of this is to come. So, by definition, optogenetics is a biological technique which involves the use of light to control cells in living tissue, typically neurons, by genetically expressing these light-sensitive ion channels. This is really cool. We can use light to control a cell. It's a really cool thing. And if you guys remember your NERTS potentials and all of this, I'll explain it really quick. So the one we'll be working with mostly is this channel rhodopsin right here. And it's sensitive to a 470 nanometer wavelength. Remember, you think the visual spectrum, that we see goes from roughly, say, 400 to 700, and this is one wavelength, plus or minus a few, that it is sensitive to. Now, when this bluish light hits this light-sensitive channel, it'll open, and it's non-selective for cations. So the net thing that will happen will be a positive influx into the cell, and then an action potential will occur. So that is how the basics of this opsin will work. So how do you get this opsin into an organism? So a lot of the mice that we work with in science are made transgenically, which means that we'll take DNA and actually put it into the male pronucleus of an oocyte, and then this oocyte will be put in somewhat of a surrogate mother, which will then produce offspring that have this transgenetic modification in them. So the mice that I'll be working with will be modified in such a way. Another way, since this is not really realistic for humans, would be using a virus, and one of these viruses could be a lentivirus. This nice diagram was actually made by Marion. So the way that this would work is that you would transfect a host cell with a DNA plasmid of desired proteins, which would be up here. Now this host cell is going to produce RNA alongside some proteins that are essential for, say, the virus envelope, also the capsid, and its farther replication. This virus is going to attach to whatever target cell you have. The membranes are going to bridge, it's going to let out the virus, it's going to replicate in that cell. Reverse transcriptase will take the RNA, make it into DNA, which will then be targeted to the nucleus. The nucleus will translate this DNA into the proteins, and in this case, it could be channel rhodopsin or some kind of opsin. And so that's another way that an optogenetic channel could be put into an organism. Right, so the question, remember what optogenetics is. Now, by my definition, I might say retinal processing is pretty similar. So it's a biological phenomenon which occurs, which involves the transduction of light to activate cells. And this is also in living tissue, typically neurons, that have been genetically designed to express light-sensitive ion channels. So you can see the similarity if you use these two definitions. And in a normal everyday situation, we might see this awesome purple car with no door handles, and the light will hit it, right? And this light will go, it'll fall onto our retina, our photoreceptors will transduce this light through the 3 layers of the retina, and if all works, we will see the same image. Now, there's a disease called retinitis pigmentosa. There's also another one called macular degeneration. Both of these are retinal diseases that eliminate the photoreceptor layer. So we lose the ability to transduce light, and without this transduction of light, you can no longer see. So in effect, this retina isn't all gone, but the ability for it to transduce light is. But what if we replace it with optogenetics? So we put an optogenetic channel in particular in this study, in the bipolar layer, so the second layer in the retina. What will happen? Well, remember I told you it only can see around 470 nanometer light. So it's not going to see all of these light waves, but maybe just one. So we're not going to see a perfect car, most likely, but maybe we'll see, a rough estimation of that car. But we really don't know. Maybe we won't see that car at all. Maybe what we'll see is something like this. And that's the question. We really don't know what we're going to see. And that's why this is so cool. This is the ability to transduce light once again, to give a mechanism to the eye, to the brain, to see again. But we don't know what it's going to see. This has never been done in humans. And so we're doing it in mice. And the mice we have are 4 groups of mice. So we're collaborating with a lab in Bern by Sonia Kleinlogel. She has developed a really, really sensitive opsin, which is really cool. And this is called OptoMBlu-R6. So they're positive for this, which is a opsin, as I was talking about, like one of these channel rhodopsins. And they're also RD1 positive. This RD1 positive means it's a retinal degeneration, retinitis pigmentosa retinal degeneration model, where the photoreceptor layer will die as it would in a human. So it's replicating this cell death, but it happens really fast, actually. This is going to be in the on-bipolar cells. And the initial findings report that the visual responses under normal daylight conditions, which is really, really huge because the channel rhodopsin mice before this, the visual responses are under extremely bright conditions. And this means that conditions such as you're standing on a snowy mountaintop with full sun coming in, or there's a laser being shined in your eye, you need really, really high light intensity. Whereas this might work with a computer monitor, which will really revolutionize how we can study optogenetics. We'll also have wild type.
Question. What's the MBlu-R6? So this opto is attached to the metabotropic glutamate receptor and activates this?
Yes, I think it works on this mechanism. I'm not entirely sure at this moment how this will work. I can definitely talk with you later about this.
Yeah,
okay. And we'll also have wild type controls who will be positive and negative for these channel rhodopsins, but also RD1 negative. So they're effectively normal seeing mice. And we'll also have RD1 controls who won't have any channel rhodopsin, but they'll be positive for this retinal degeneration, and therefore they'll effectively blind. So we'll have these 4 groups. And now that we have our mice, how do we know that their genotype is actually correct. So one thing we can do is optical coherence tomography. And what this is in effect is a technique for obtaining subsurface images of a translucent or opaque thing, such as an eyeball. So it's effective to a low power microscope, some kind of optical ultrasound if you want to say. This uses near infrared light, interferometry. So shine the light, it comes back, and you can kind of measure the tissue with cross-sectional images. So this would be an example of a wild-type retina, right? It looks healthy, it looks nice. And we actually did this. I don't know if I mentioned that. So this is a wild-type retina in one of our mice. And this is the RD1 retina in one of our mice. So you can see there's a lot of cell death, a lot of degeneration going on here. And I actually made a little video. So this is a video going through the retina. On the left you have wild type, on the right you have retina degeneration. So just look at the thickness of the retina. If I stop it at any time, you can see that there's a lot more thickness of the retina on the left than on the right. And this is about 200 micrometers scale. Oh, that's the next video. Okay. So now that we know we have our mice, what are we gonna do? How are we gonna figure out what they can see? So I want everyone to take a moment right now and imagine that you're walking in this room for the very first time. Now, there is no captivating talk happening, you're all alone, you're just walking in, and you're looking around the room. You see these white curtains on the left, you see these windows on the right. You're in here for, say, 30 minutes with no task. I just put you in this room. Now, the next day comes, I put you in this room again. For 30 minutes, you walk around, no task. You're just looking at the room. You're really starting to get familiar with it. 5 days of this, 30 minutes, 30 minutes, 30 minutes. You understand this room, everything in it, right? Now, that is what I did with the mice. So this is their room. It's pretty plain compared to this room. You have some black walls, a separator. This silver area, this gray area, is where they can walk around, essentially in 2 zones, and they can go back and forth between them. This and this are both monitors. Now they're on, and they're black right now. So there's probably a little sound from the monitors being on, maybe a little dim light, but not too much light. There's also a lid going to be placed on top of this box. So they're pretty familiar with this, right? Now day 6 comes along. Think about this. Day 6, you come back in your room, you're walking around this room. Now 8 minutes into your normal 30-minute routine, I push a button, and boom, all these curtains turn black. What are you gonna do? You're gonna be pretty excited, right? I would get my heart pumping. I'd be, whoa, what's happening? So what it seems to happen with the mice is that they run more. They run. They get excited about this. So these turn to white, and they just start running more. So what happened right there is that something in the visual environment changed, and it produced a behavioral response that we can measure. So that's pretty cool. So now that I know that this, say, happens in wild-type mice, I can try it in channel rhodopsin or optogenetically treated mice and see if it also is there, and if it is, I can be confident that perhaps they can see this as well. So in essence, I'm taking 3 blind mice and seeing how they run. And as I said, compare the behavior immediately prior to and after that stimulus. So the things that we can look at are distance, like total distance ran, the time they spend in zones. So I could, for instance, only turn on one of the monitors. And if the mouse, say, stays in this side more, maybe he wants to explore this, whatever this new visual thing is. Or maybe he wants to stay away from it. So this is something else that we can measure. We also measure the path. Maybe the mouse normally runs around the edges of the box, but when the monitors are on, it wants to stay close to them, or it wants to stay far away from them. So these are all things that we can measure. Just to take you through the box really quickly, this was one of the drawings I had of it, because I don't have a full picture of it, unfortunately. But I'll take you through it. So 2 monitors in the end wall, which you saw. Say 1 monitor, 2 monitors. There's 3 divider walls. There's 1 here, there's 1 here, and there's 1 here. So it separates the box into different portions, which we can do different things in. There's zone 1 and zone 2, which are effectively just this side and this side. There's an infrared camera right now that's mounted from the ceiling on top of the box. The top of the box is red plastic. So mice can't see red, but but the camera can see through the red, and we have infrared light shining, illuminating the mouse. So that really helps us track the mouse. There's an escape habitat, which I will show you guys in a moment. And some of the program stimuli we have are dark light, full field flashes, flickers, drifting gradients at any visual acuity, rotating stripes, we can change the contrast. And we also have something called the looming stimulus, which Thomas and also Marcus Meister and Yilmaz from Caltech and Harvard really investigated. And think about this, if you're a mouse in a field, right? Nice blue sky above you. What are you're worried about getting eaten by a bird, right? So you're walking around and this bird is up there, it's a little black dot. Now as the bird sees you and he wants to eat you, he comes closer to you. So in your visual field, there's going to be something coming, it's going to get bigger and bigger and bigger. So this can be replicated by making a black disc just expand and expand and expand, and this replicates the shadow of a bird coming closer to the mouse. And this monitor is in the ceiling of the box, so we can put the mouse in there and replicate this. And I want to show you what this looks like. So mice generally have two responses to this. They'll either freeze, and the reason they'll freeze is because if if a bird was seeing a mouse walk in this grass, right? The grass is all stationary, but the mouse is moving. So maybe the bird is seeing the mouse because the mouse is moving. So if the mouse freezes, maybe the bird loses it. So that's one behavior. The second behavior is escape behavior. Maybe it just wants to get out of there. So what we've provided is an escape habitat. This is something that this mouse is used to. It's been in its cage. It's made out of red. So again, it thinks it's solid, that no one can see it. And when you see this whole section flash, light up, that means the stimulus just happened. The mouse is going to go off screen, but that's okay. So there's the mouse, goes off the screen. Flash happens, and he runs immediately into his habitat. Pretty cool. So this is not only cool because it worked, but it's super cool because it shows that the mouse integrated a visual response into not only a relevant behavior, but a survival behavior. So if we can show that this works also in the channel rhodopsin mice, in the optogenetically treated mice, think about it. That means that they can still see some visual stimulus well enough that it gives them a behaviorally relevant response to their lives. And all of that is tracked in real time, which unfortunately I just forgot to show you the video for that. So this is real-time tracking going on. So you can see the mouse is red right now. These are infrared lights on the right, so he can't see them. The mouse walks around, it's being tracked. Sorry, this is my cell phone camera, that's why it's so bad. And that's what that looks like. All right. Yeah.
So was the video that you showed a healthy mouse?
Hmm? Yeah, I've only worked with the wild type mice so far, and so in the near future we'll be working with the optogenetic treated mice. So just for some preliminary results of what I found so far. So this, there's a lot going on here, and it looks pretty bad, but I'll explain it to you. So this is first on the x-axis. This is 3 minutes of time, binned in 30 seconds each. So there's 6 bins, 30 seconds each, that's 3 minutes. The first 2 bins represent 1 minute of control, right? And they've been in the box now for 7 minutes, and this is the 1 minute before the stimulus. The stimulus onset is here, 1 minute of stimulus, and then 1 minute after the stimulus, which is in effect visually the same as the control, it's just after the stimulus. Okay? On this axis, I normalize the amount of time that the amount of distance that the mouse ran. So the mouse ran some distance over this 3 minutes, and I take this 30 seconds and divide it by the total 3 minutes. I did this because mice run at different, some run a lot, some don't run a lot, so the data points would be all over if I didn't normalize them. So what this looks like, what's really important to look at is just in between these 2 bars right here. So you notice this is the last 30 seconds of control, it's the first 30 seconds of stimulus. And all of these slopes, except for one, are going up, which means they increase the amount of time they ran coming onto the stimulus. And this was significant, but this is just preliminary, and we need to look at this more in depth. I did this again, and got these results. Looks like all of them are now increasing, and then I put these together full field. So it seems like the full field does really generate motion, which is good to know. I also did a flicker, 4 Hz flicker of one screen, and this is what happened. Significant. Did it again, still significant, put them together, still seems significant. 10 Hz also seems significant. I also did moving stripes, so this is just drifting gratings at a speed of 0.5 cycles per second. This not only wasn't significant, but it also looked like it maybe, I don't know, if I did it a lot more, perhaps it would make them run less. Interesting. Sped up the cycles, and now it just looks like it's all over the place. So those are the preliminary results. So what's next? So some cool things that we can do is first something called the drum. And this is the Optokinetic reflex drum. And this is designed by Boris back there. Also Garrett Ecke, who I worked with in my last lab rotation, Marion, and also Thomas. And this is pretty cool because it uses a standing platform with auto-adjusting stripes to keep a constant visual acuity. And what that means is that these stripes are going to be a certain distance apart. Now, as I get closer to them, they're going to take up a wider spot on my visual angle. Yeah? Okay. They're going to take up a wider spot on my visual angle. Now, what I can do then is make them narrower as I get closer to them. So the visual angle of the stripes is always constant. And because of that, we can really finely see what they can see. Because in this, when the stripes move, so the stripes rotate around the mouse's head follows the stripes. This is a reflex that the mouse has. And I will show you what this looks like. So there's the mouse, there's the stripes. You can see its head following the direction of the stripes. Great. And again, this is cool because we can make the stripes so narrow that then they become gray, right? And then the mouse will not have this head motion anymore. So we can really see how fine can this mouse see. Something else we can do is a paradigm developed by Laura Bousset and Stefan Katzner, which we're going to call the disc. This disc was created by Hans-Jürgen Dahmen, who I sat across from in my last lab rotation. Really cool guy. And they taught mice many, many different things with this. They did orientation discrimination, shapes, contrast, a lot. And what's great about this, so the mouse is head mounted, right? And it's on a disc, so it can run in place freely and the disc will move. There's a water dispenser to give it water, and right in front of it there will be monitors that display some kind of stimuli. The great thing is it's a quick learning time. It only takes about 10 days for most mice to learn different things. And we can learn finely tuned things. And what we're thinking about doing is orientation discrimination. So if I teach the mouse that bars going this way are rewards and bars going this way aren't rewards, so I show them this one and then say half a second after presentation, I give him some water. And he licks for the water. When I give him this one, nothing happens, right? Over time, water, 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 no water, no water. So what the mouse will start to do is anticipate when I show him this, because it's going to be a little after, half a second after, that water is going to come. So he'll start licking. And cleverly, there's a lick sensor on the end of this water spout. So we can measure the amount of licks he has when the rewarding stimuli comes versus the non-one. And amazingly, this seems to work great. So this is something that we can also do. Lastly, once the mouse has passed away, we can do some multi-electrode array in histology. We could take the retina, put it on a multi-electrode array, show it different visual patterns, and then measure the responses to these visual patterns. And we can also do histology to see how well the opsins were taken up into those cells. And lastly, I have some questions for you guys. You don't need to answer them now, but maybe think about it. We can talk another time. But what stimuli are uniquely constructed to suit a visual paradigm such as this? So think in which features would be most interesting to explore the perception of optogenetically treated mice. So right now we're looking at orientations, contrast, shapes, flickers, movements. But what basic visual features would be interesting to see if this mouse can see? My second question would be, how can we improve upon these methodologies to better realize the goal of vision restoration? So can we combine opsins, use channel rhodopsin, excitatory, and also halo rhodopsin inhibitory, and make a push-pull mechanism or anything like this. And also engineering new opsins to have greater wavelength ranges and things such as this. That's it. Thank you. References. And lastly, this is the lab. And although I'm not in this picture, I'm climbing my way there.
Okay, Eric, thank you so much for your talk. It was very interesting. Does anyone have any questions?
Barbara?
Do you think theoretically it's possible to restore color vision using a technique like this?
I don't think it's possible in the way that we're doing right now because we only have one opsin, and that one opsin can only receive one wavelength. And so I don't know how we could selectively gather enough information from the light to then separate it into colors. But if we had different opsins, maybe. Cool.
When you presented the full field flashes, did you try different intensity levels?
Not yet. Most of the time that I've spent in this lab rotation so far was just preparing everything, and I just started collecting data, I guess 2 weeks ago. So I've done 8 days of data collection so far. So I haven't done different intensities, but I've done different hertz of flickering, so fast flickers or slow flickers, but not the intensities yet.
So the mice tested so far, they all lost that, right? So you expect